What I'm going to talk about today is essentially the same talk I gave at the International Genetics or International Congress on Quantitative Genetics. So if you were there, feel free to get up right now and walk out. I will not be offended. But I hope to end here with a little bit of discussion on this, and maybe we could, may not have time all today, but over the next month or two, as people run into one another, I'd love to chat more about some of the ideas in here, because I'm not fully confident that what I've got here is correct, but it's an opportunity to chat about it. And when I am standing here telling you about the work that we've done, I want to remind everybody that really this has been a tremendous collaborative effort for over a decade by this great group of collaborators, including Mike McMullen, Sherry Flint Garcia, Jim Holland, Doreen Ware, Chi Sun, Steve Kresovich, John Dobley, and the USDA, ARS, and NSF have helped fund this. Okay, so like many of you, we want to create this global model of genotype to phenotype. And I'm going to put up the genomic selection worldview that if you're an applied breeder, you're going to say, this is crazy, okay? But as a geneticist, this is the way I kind of view it. So this is our normal breeding cycle in maize. During the 20th century, maybe it took 5 years to go through that cycle. The cycle that we can do now is every 4 months. We can make a cross every 4 months, and some companies say they can push out actually 4 generations a year, so it's down to 3. And so, of course, what we want to do is if we could predict that phenotype perfectly, then we could theoretically, make breeding move 15-fold faster biologically. Now we know, everybody here knows the accuracy of our genomic selection predictions are nowhere near that good, and we can't get to that type of reality. But maybe right now we can push 3. But essentially, the thinking behind my group's work is, okay, this is what we can do with a fairly naive model. What can we do if we really were able to predict as well as we could by looking at phenotype? And then we'd be pushing essentially the biological maximum, which would allow us to breed at 15 times the rate that we do currently. Okay? So that's what motivates a lot of my thinking in the group. The second thing that motivated me is essentially how much natural variation is present in maize. It started out with a lot of these studies in evolution, but I'm just still shocked that 2 corn plants are as different as humans are from chimpanzees. And that does mean that there are hundreds of thousands of functional SNPs that are segregating in the genome. And so the challenge that we have is to then make those connections between the hundreds of thousands of functional SNPs and the phenotype. The second thing I always want to remind people, and this is probably right now still probably our biggest challenge genomically in maize, is dealing with the nature of the genome. If you sequence 2 inbred lines of maize, some pioneering work by Hugo Duner about a decade ago showed us that essentially they only really line up in the same homologous base pairs at the same position about half the time. And there's been this rich history of retrotransposons that move things around, genes and everything else. And from a technical point of view, that's probably still our biggest obstacle, is dealing with the fact that non-collinearity of the genome, because we now also know that a lot of these regions in between the genes are very key for controlling complex traits. And when human geneticists always talk about, oh, we have all these copy number variants between 2 individuals, I just laugh, because there's just virtually nothing going on in the human genome. The last thing that was interesting in a vertebrate genome in terms of dynamics was the Alu explosion about 65 million years ago. Okay? So but you may start asking, why is it like this? And essentially, it's just because a lot of our plants, including maize, have fairly large effective population sizes. And some of that coalescent and some of those alleles probably start diverging from one another older than the divergence between Zea and its sister genus Tripsacum. They even have different chromosome numbers, but we find shared polymorphisms between them. And some of that variation probably really dates back all the way to the Pleistocene. Human genetics, none of us are probably really that accurate in our clocks, but we know that human genetics probably only dates back about 20,000 generations. So we're talking just about the magnitude, and a lot of this difference in the genome structure just comes from the fact that maize has a coalescent that at some places in the genome stretches back multi-millions of years. Not all of it, but some of it does. So this is where we get to the question that's I've been pondering a lot the last few years is, how does this effective population size and the census size vary through time? And please read this thing here. The numbers in green are totally made up, not totally made up. They're an educated guess, okay? But let's, if we start playing with some various numbers here, we've gone through all this fluctuation. And we have here our effective population size. And then we also have the census numbers. I mean, we really do know. We've got 10 trillion corn plants that are grown on the planet each year. Okay, that number we actually do know. And then, but through the rest of these, we don't really have a great idea where each of these fall. But you can see it's fluctuated tremendously through time. And whether we're talking through domestication, land races, modern maize. But also we have some very large differences between what the effective population size is and probably the census size. And that does mean, though, there have been organisms out there exploring the landscape, adapting to different environments. Many of them, of course, those experiments go extinct and don't pass anything on. But the population, those population sizes have been essentially what's created that genetic architecture as it evolves across a landscape. And one other thing I want to kind of highlight is, as we go through this, I actually think that someday, and this is, totally, we're not sure that it's true at all, is whether or not Tripsacum really held a lot of the key ancestral variation, and all of Zea is essentially just one little offshoot of Tripsacum. Tripsacum is the dominant genus over the hemisphere, over North and South America, and it may well be that Zea is the little offshoot. So I'll be very interested to see how much functional variation eventually someday we can attribute to Tripsacum. So maize genetic variation has evolved over a wide range of contexts. And you can just see, we've gone back and forth. Notice these are log 10. I mean, we were talking massive shifts in effective population size over time, and always this big gap between the census population and the effective population size. But I also want to highlight that what we think, we of course don't have a really good idea what happened during the Pleistocene, but we know some of the very biggest shifts have all happened really recently. And I think that's what's also really, as I'll get back to at the very end, is contributing to the genetic architecture that we see today. Okay, so I'll go over some of those tools that we've been using for trait dissection that we and much of the community have worked hard to put together, as Wojtek was mentioning. Then I'll talk a little bit about the genetic architecture, starting with simplest, going to complex, and then if we have time, talk about some of the challenges. Okay, so essentially the scheme that we've been working towards is of course, we want to know the genotype of everything. I mean, that's, if you know the genotype of everything and you know the phenotype of everything, it makes it a lot simpler to draw the lines between those two, okay? So that's the basic scheme. Of course, we had the reference genome being sequenced. And that's been a tremendous aid for doing everything. Our group has helped lead a lot of this whole genome resequencing. And then we've also been doing what we call genotyping by sequencing. And essentially, between these three approaches, we're then trying to come up with an estimate of what the genotype is of all this diverse maize. I'll give you a little feel for the HatMap project. It just came out this summer, but it was through a great group of collaborators led by Doreen Ware's group at Culturing Harbor, Jeff Rossabara at UC Davis, and colleagues at the Beijing Genome Institute. And Jeremy Chia at Culturing Harbor really is the one who led the analysis and did a great job of it. So to simplify a heck of a lot of work, we identified about 55 million SNPs. And the key part of that is that gave us now SNPs that were in strong LD and strong correlations with about 80% of the genome. When we only had about 1.5 million SNPs, we were only in strong LD about 30% of the time, so it's a big jump for us. We also were able to identify copy number and presence-absence variation across the genome, and we can now show that 80 to 90% of the genome, if you go just region by region, shows significant evidence for copy number variation. Essentially, the only thing that doesn't are a few highly conserved genes. Everything else is showing some presence-absence variation in some line. Okay, the next tool we use is genotyping by sequencing. Obviously, here in Cornell, a lot of discussion of this. Don't need to say too much about that. Rob, of course, got this all kicked off for us, and actually Mike Gore also got it kicked off with the first restriction map. So you guys know how this works. Cut up the genome, read things out. Yeah. Okay, now Sharon's not here. Okay, I actually changed this slide just for Sharon Mitchell because she hates it when I show this slide. Okay, so what's the cost of this? And this is the molecular biology cost of doing this whole thing. And it really, it really has allowed us to survey the genome much more cheaply than it was before. When we got started a couple years ago, genotyping lots of material, the Illumina 55K array was costing about $200 or $300, so it was per sample. So it's got, now, of course, with price pressure, it's going down. But I do want to now make this warning for Sharon's. The cost of the bioinformatics is substantially higher than the cost of doing the work and everything. So, and it is getting easier, but it has been fun. So, okay. So what, Rob, you disagree with it being fun?
Mark just asked me if I was having fun, and I said I'm having a lot of fun.
Very good. Okay. So what we have right now in our group, and with lots of collaborators, is we've genotyped about 30,000 lines of maize. About 14% of the tags come from these regions that are too repetitive. Lots of that map frequently. We call about 680,000 tips frequently, about another million at low coverage, and then just millions of these presence-absence variants. That if you want to learn more about, Faye Liu can tell you more about that. But essentially, this gives you a little key for what we can start looking at right now. And it's everything from breeding lines to land races and inbred lines. And we're essentially able to really start discovering alleles and polymorphisms at that level of down to 0.1% frequency. And so we're able to start avoiding the biases that we had by having a small subset where we would only sequence 20 individuals. And then all you're doing is rediscovering the SNPs from those 20 individuals. So it's been exciting to be able to do that. And now, because of the work of lots of colleagues, essentially all this material is being replicated in lots of plots and evaluated around the world. Second tool we created was NAM. You guys all know what this is. We took 25 lines, crossed them, created 5,000 inbred lines. All right. Did anybody want to hear more about NAM? All right. But I do want to highlight why, scientifically, why NAM is useful. Okay. The first thing is we spent a lot of time working on the statistics of association mapping, and through the mixed model approaches with Jiaming Yu and Jiwu Zhang and the group have really helped make that kind of work. But sometimes we hit a roadblock. There's just, if the population structure and the trait you're interested in coincide too much, there's just really not we can avoid false positives, but we don't necessarily have a good way to get good statistical power. At some point, you need to do genetics. And that's what NAM does. It does genetics. It makes crosses, breaks things up, allows us to look at that population structure. We can project the SNPs accurately. We have almost in that short period of time, virtually no mutation. And one of the key things that we've been able to do, and Peter Bradbury has really been doing a great job of leading, is how do we partition out that genome? And so we really now can control for almost all the genetic variation on the 9 chromosomes we're not caring about at one moment, and then just do GWAS on the target chromosome. So you're able to essentially genetically control 90% of the genetic variants through linkage, and then just do association mapping on the remaining portion. And then, of course, we can test out a wide range of results. And in the end, we can also compare how well we're doing between linkage and GWAS. Are we explaining the genetic variants that linkage mapping tells us is present there? Yes or no? And that really gives you a real concrete answer as to whether or not GWAS is working. So we've also been creating lots of association panels. Some really great statistical approaches have been developed over the last couple of years. And now we're being scaled up to where we can work with thousands and tens of thousands of individuals. So, okay, so let's now talk about some of the simpler traits. I'm not going to tell you too much about, we did provitamin A, Torbert, and Mike have already told you about this. Okay, the key thing I want to highlight here is that, this really was a pretty tractable trait. Essentially, alleles of 2 genes allowed us to put the right ones in, allowed us to increase beta carotene, gave us a 16-fold increase in beta carotene. But one of the key things, and I think is always important to let people know about, is that the 2 best alleles really weren't in the same breeding pool. If you created, that breeding effort, even in something that looked pretty diverse, just out of one population, the alleles just weren't there. The 2 best alleles weren't there. And so it really did having a directed approach that said, hey, the one set of alleles is over here, the other one's over there, let's mix these together, really did help push this forward. And then through Torbert's work, working with Kevin Pixley and Simmet, they now have already, this variety, first cycle is in feeding trials this year, and in 2 years from now, they hope to have one much better. But we'll learn more about that hopefully later. So Alex Lipka has been now leading an analysis with Mike Gore on doing GWAS of the alpha tocopherols. And what I want to show, with a big enough gene, big enough effects, we can get big hits, okay? But what we're also seeing is often in this case, we only get one hit with an association panel. Now when we take that same type of traits, we hit the tocochromanol compounds, we hit it with NAM, we see 10 times, 11 times as many genes controlling the trait. Essentially, on a lot of these small association panels of a couple hundred, you're just totally underpowered to see the true genetic architecture of the trait. One of the most exciting things that I've enjoyed working on this trait, or these traits with Torbert and Mike, is being able to compare the results for these 2 tractable compounds with the more complex compounds. Because we know in NAM, essentially, these are, we have massive amounts of statistical power for these 2 simple traits, okay? If we only can find 5 or 7 QTL, that's probably as many QTL that really are controlling it, okay? In other traits, flowering time and things like that, we essentially are exhausting our statistical power at some point. But both of them are quite tractable, and we see essentially for traits that are equal heritability, a very big difference in the number of genes that are controlling them. The tocochromanol compounds essentially are 2 to 3 times as complex in terms of number of genes as the carotenoids are. And so the model, and we'll then keep on going back to this type of model. I showed you how the effective population sizes are varied. What I want to highlight is that carotenoids, especially the seed color, the one we're scoring here, were only visible and could only be the target of selection starting about 8,000 years ago, once the gloom gets removed. That trait is only 8,000 years old. Tocochромanols are the key compound that prevents the lipids from oxidizing in the seed. And they're really key for a seed being in a seed bank. And so the natural history through time has probably been dramatically different for these 2 pathways and allowing the genetic architecture to evolve for them. And so I think the reason we had trivial genetic architecture, really simple one in carotenoids in the seed is because it's young, tocochromanol because it's old. Okay, let's step up to something a little bit more complex. We're still looking at metabolites, carbon and nitrogen metabolism. Nengyi Zhang was doing this, and this is a great collaboration with Mark Stitt and Yves Gabon's group. The key thing is we were able, with their help, be able to look at 12,000 field sampled plants. And then Nengyi did a great job of factoring out all these factors like maturity, weather, position, lots of other confounding factors. Able to look at what genes are being controlled, pathways, and essentially what he was able to see is that at this level, and I'll show a contrast with this as we go up in a more complex trait, the correlation between 2 traits really was based on a correlation of having the same QTLs, okay? And as we start going up the genetic architecture, you'll see that this starts disappearing. So what we're seeing is lots of pleiotropy at this scale. And one of the key genes that he hit And now groups like Tom Brunel's are now focusing on is, for example, he hit a direct hit in the key C4 gene, carbonic anhydrase, and that gene turned out to be controlling a substantial portion of this pathway. And for those of you who aren't familiar with it, in C4 plants like maize, the CO2 comes in, it's converted into using carbonic anhydrase to carbonic acid, and then it's pumped into the bundle sheath cell with things like malate transporters helping push it in where carbon fixation occurs. And so this really is probably one of the key genes that is determining what that carbon balance is, at what time, and at how much you want in each leaf and everything. Because once you start pushing the carbonic acid through, you're set, or the die is essentially cast here. And as we try to make C3 plant into C4, obviously getting this tuned exactly right for the right environment is probably going to be really key. Okay, so let's start moving into some more complex traits. And there's been lots of great efforts done this by lots of colleagues, and I'm going to just summarize pretty much tons of work in one slide, and many people here have contributed. Maps everything. And then for everything, these essentially you can kind of envision, these are all the traits that you could see with your eye, okay? If you could see it with your eye, a ruler or something like that, this is the summary. Everything was controlled by lots of QTL, mostly additive. Prediction works fairly well in terms of it explains most of the that we can explain most of the heritability. And we hit genes reasonably well. But there are some big zones where we have low recombination and it doesn't work as well. The classic example of how small the effects were flowering time. We started out with parents that differed for flowering time by over a month. And our biggest effects turned out to be about 36 hours. This is a single gene called ZMCCT, and it produces 36-hour effect. So essentially, the reason why we have very different plants that flower very differently is that late flowering plants have lots of late flowering QTLs, and early flowering ones have lots of early flowering ones. And so it's a very simple Fisherian model of what creates variation. Pat Brown, some of you are probably now too young to know that Pat Brown was here. But Pat Brown was a student of Steve Kresovich's and then worked with us and the inflorescence project for a while. And he did a really nice study that came out in PLOS Genetics last year. And what he did is he directly compared, standardized everything for various traits, and then looked at allele frequency. And I want you all to carefully look at this diagram. And we have two traits, tassel, and he had lots of tassel traits and ear traits, maybe 5 or 10 of each of these, and then various flowering and leaf traits that we've measured. And I want to highlight that essentially, pretty much at each step, the scaled effect sizes of the tassel and ear ones are larger, no matter what the allele frequency, than the ones for leaf and flowering architecture. So essentially, we started out with roughly equal heritabilities for all of these. It's the exact same germplasm, and it looks like these tassel and ear traits, and what I want to highlight is these traits don't really exist. The ear of teosinte inflorescence really, why it took so long to figure out, it really doesn't look at all like a maize ear. And when you talk about row number, there's only one row number in teosinte. It's 2. There is no variation for that. I'm sure you could find a mutant that's out there, but in general, there's no variation for it. And the tassels have also dramatically changed. They're in a different place and everything. So and so you can just see these effect differences have changed. I'll get back to that in a second. But essentially, we also have been able to see using NAM that if we add up the effects across all these individuals, we can predict. But I want to highlight what this is really showing is that there's little epistasis that we can't deal with. It doesn't mean that we can predict across all of Zea with this level of accuracy. Essentially, you can kind of imagine we've taken 5,000 lines to try to figure out if we can predict 26, okay? This is not what you want, a cost-effective way to do prediction for genomic selection, okay? So I also want to say, indicate that we've been able to get similar types of results with some less power, but sometimes some better resolution using some large association panels. Cinta Romay has put together an association panel with the folks at in Ames, Iowa, that included essentially every inbred line that are in the USDA stock center. She genotyped them with GWAS. And at this analysis that she did with 680,000 SNPs, we knew about this one gene, VGT1, and this one gene, VGT1. It shows a lovely, what we call a skyscraper. You see it, we hit it. But I want to highlight, this is this GHG7 gene, that is also known as ZMCCT. That gene, we only hit it with a single polymorphism. And we know from NAM, it is by far the largest effect flowering time QTL. That's the 36-hour one, okay? That's the one, if we were to do association mapping, we should have been able to easily hit, okay? We just got lucky. We actually missed that SNP when we have 1.5 million SNPs in HapMap1. And now, once we get to HapMap 2, where we've got 55 million SNPs, then we have about 15 or 20 SNPs that are finally in LD with the causative site. But essentially, because the effective populations, what we're seeing is the difference in power caused by effective population size. VGT1 became common and was important for the adaptation of temperate germplasm. It's gone through a severe bottleneck. Much smaller effective population sizes. Northern Flints are probably founded from 1,000, 2,000 individuals that then made it into North America. GHG7 is most common in tropical areas. Effective population sizes there were 2 orders of magnitude larger. You need 2 orders of magnitude more SNPs to go figure out anything in tropical corn than you do in temperate corn. Okay. So we also, through some nice work done by Peter Bradbury, he used HapMap2 and presence-absence variation and was then able to look at how often various classes of markers were enriched. And we were able to compare HapMap2 versus read depth variance. And in terms of enrichment, the HapMap1, which was looking at genic, were definitely enriched, and the read depth variants were also highly enriched, in some cases like almost 20-fold enriched. And when we looked at everything overall, what we're seeing is essentially, even at these preliminary stages of our ability to be able to quantify copy number variation, sometimes more than 1/3 of all the QTL were being explained by a copy number variation. The methyl filtered variation, this is the variation that's coming from the genes, mostly. And then a lot of these HapMap2 ones are just a lot of those are intergenic. So I suspect, and we can't prove this yet, but I suspect once we are able to really quantify all that variation intergenically in the presence-absence variation, we'll hit 50% of the GWAS will be, the phenotypic variation will be coming from intergenic regions for adaptive traits. Okay, so let's get back to the thing, or let's talk a little bit about genetic overlap and genetic architecture between traits. Feng Tian did this very nice analysis, and one of the things, this stands in contrast to that metabolite analysis that Nengi had done, where we saw lots of pleiotropy between all traits were controlled by the same set of genes. When we start looking at a trait you can measure with your eye, I certainly expected some real genetic architecture between leaf angle or leaf length and leaf width. Now, some of my developmental biologists said I was stupid to think that way, but it's the leaf, it comes in wide, and I don't know. I thought they would be really shared. But I'm still shocked that only 2 out of 35 genes or loci seem to be shared between those two. The only trait that we see that shows substantial pleiotropy is developmental rate or flowering time. So everything else really shows fair amount of modularity among the developmental traits. And there's relatively little genetic architecture overlap. Okay, Jason Pfeiffer has looked at height And this turned out, again, this is one of those traits that I thought was going to be roughly equally complex with flowering time, but that turned out to be wrong. Essentially, the biggest estimated effects that we were seeing here were essentially about the order of 4 centimeters. I mean, really tiny effects. When we've done fine mapping, it really confirmed, again, these very small effects. But when Jason was able to add up all of these small effects, prediction was still pretty good. And he's been working on applying various genomic selection models to these. And he is getting out fairly accurate estimates because we do have enough individuals to start really training on. And then we also observed that there's this set of outliers. And we're still kind of arguing over what these outliers really mean. But there's a group of lines, not a very large percent, 1 or 2%, that essentially the prediction isn't holding up. Okay? It turns out that these plants are all shorter than what they're predicted to be. Okay? Is that a novel mutation? Jason also knows that a lot of these just actually get thrown out of the field trials more often too. Okay? So it's not clear whether or not this is a phenotyping error or these are just bad lines that are carrying load all over the place. Why are the field trials for those set of lines failing more and more often? Okay? So these are the type of questions that I think we're now getting. We've got the big picture of some of these common alleles kind of figured out. We need to start worrying about our outliers. Why are our outliers there? I don't know yet, but we're going to be working on that. So in terms of complex developmental traits, kind of what we're seeing is ear morphology, again, is relatively simple, and I think that's because it's only essentially a 10,000-year-old trait. Flowering time probably goes through various oscillations in Mexico. During the Pleistocene may not be quite as old, but height for some reason seems to be even more complex than flowering, and that would suggest to me that perhaps some of its genetic variations going back even farther, and maybe that's just because of competition with weeds and stuff like that is something that's ever present. Okay, let's now talk a little bit about heterosis. One of the analyses that just fell out of the NAM experiment was essentially we carried on 5,000 parallel inbreeding depression experiments. We took 2 parents, we crossed them, we selfed them down. And we expect at the end in each individual, about 3.5%, 3.2% residual variance or residual heterozygosity. And instead what we saw was that, the centromeres or the arms were roughly at that level, but every centromeric block was higher for every single chromosome. And as we started getting a feel for what the genome looks like, this is a recombination map. Mike McMullen helped us put together the first one in NAM, and then Peter Bradbury has worked on a second one using GWAS markers, so now at very high density. And you can see that there's this massive drop in recombination. These are the 10 chromosomes of maize, and a massive, many orders of magnitude drop in recombination near the center. So, and what it turns out is that there's actually a fair amount of genes stuck in these. Initially, I think a lot of us thought there was no genes there, and so it didn't matter. But it turns out it has less frequent genes, but there's still a reasonable number of genes. And so one of the analyses we were then able to do is then essentially plot residual heterozygosity against the inverse in recombination. And we've got some chromosomes that have some really strange recombination patterns, and you can see the residual heterozygosity follows it beautifully. And essentially what it looks to us is recombination explains about over 30% of the residual heterozygosity, and some of the other things we thought were more important, or could have been, like gene density and diversity, don't explain for very much. In recent work, Chris Schoen has also been reanalyzed 3 heterosis studies, saw similar work. Alain Chogaset had a beautiful, last year he gave a beautiful talk on this in the seminar series. And essentially we are starting to see this consistent overlap in QTL locations. And later this semester you'll hear from Sarah Larson in a direct NAM comparison of this. But the model we're now working on is essentially restricted recombination gives rise to Hill-Robertson interference, which is essentially, you can just think of it as the population genetics version of repulsion QTLs. And there's certain regions of the genome that you can't get the best alleles linked to one another, and so that then produces pseudo-overdominance, which on a genome-wide scale produces heterosis. So it's one of the oldest hypotheses for heterosis, but our data is completely consistent with that. The problem is 31% of all the genes in maize are in these regions, okay? And they still have a lot of genetic variation. And essentially, I would argue we don't really know what the breeding values are for these regions, because most effective population sizes of breeding programs, looks to me, it's about 15. And there just isn't enough mixing of all those alleles to really test them out. Okay, so I think, but there's a real simple answer. We can select for paracentromeric recombinants and then put things out. And the key thing is then we put them out in the field and evaluate them. We don't put them out in the field first. We have to select, use markers. So what we're kind of seeing here is a genetic load probably doesn't go way back, but it certainly can probably persist for a few thousand years, maybe tens of thousands of years. And modern breeding, though, has essentially organized that genetic load in a way that deals with heterosis. And that's just a very modern thing that's essentially working at this massive block scale. Okay, so I would say what we've kind of seen is Fisher's pretty much right. We can additive, modest epistasis, little pleiotropy. Trait age and effective population size seem to matter. Intergenic regions in C and Vs are very important. And I think if we get the right training data sets, we can make some useful predictions. So this is the kind of plot as I start working with the collaborators in various traits, start thinking about is, of course we would expect the genetic architecture or QTL complexity to be positively correlated with biochemical complexity. I mean, that's, simple biochemical trait should have simple genetic architecture. But the second thing I would suggest that people should think about is that age of the trait. And here we're talking about how long has selection been able to adapt it to local environments? Is it under diversifying selection at various periods in time? And I don't know there's a simple way to quantify this right now. But I think this is, and when I start looking at this type of plots, if I look at thinking these two axes, it really does start to make some sense. Now the problem is when we work on yield, it's going to be essentially culminating, of course, it's infinite biochemical complexity, but it is working with component traits that are essentially all sorts of different ages. And what may be right for one type of trait may not be appropriate for another type of trait. Okay, so I guess a couple more minutes here. So I want to talk a little bit about the challenges that we kind of see going forward. And I think, everybody gives me a hard time. I say genotyping is essentially free. And in the big scheme of things, yes, it is now approaching that limit of being virtually free. Our big problem, of course, is phenotyping. And so then I think really the question is, how can we let genotyping be that intelligent guide that allows us to figure out what to phenotype? What's the best way to select those training populations? Of course, John Luke's group is thinking a lot about that. But I also think I would ask that people start thinking about this one. When you do your simulations, let's start playing around with different ages of traits. What do different coalescence look like in setting up your ability to train on a GS model that goes many meioses? If you're only going to try to predict 2 or 3 meioses out, it probably doesn't make any difference these types of things. But if you're going to try to make a prediction that's going to work across germplasm, does the age of the trait and historic NE matter? And because eventually, as an empiricist, I want John Luke to tell me whether or not I got to put out 1,000 things, or I need to put out 100,000 things to do the training population to predict 10 meioses away. Do you have the answer already? Okay. All right. The second thing I think we're all going to just have to work on is how do we just scale up to the data scale. And for the graduate students out there, learn how to program. You got 12 trillion data points. It's hard to manipulate these things. And that's going to be life. And then one of the other areas we're looking at is rare alleles. And the purpose of breeding, of course, is to find good rare alleles and increase their frequency. Well, of course, the opposite side, of course, is to purge the bad load alleles. And so one of the things our group is going to start working on, and our maize diversity group hopefully will be able to work a lot on this in the next few years, is to try to really start working in this area. Because the problem is there's way more bad alleles out there than there are good alleles. I mean, there's probably 100-fold more bad alleles segregated in any one moment than there are good ones. So picking out, and then there's a vast number of neutral ones. So when we're going for the good ones, it's this small little, 0.1% of all the genetic variation out there, or even less than that perhaps. And a percent or two of all the variation out there is probably bad. And so the challenge will be, how can we use not only genotype to phenotype, but perhaps, and what we think is a real key one, is a lot of informatics, genomics, to help us start making some predictions on what are both good and bad alleles. Okay, and then I think the second or final area I'd like to mention is I think we're at this incredible point in genomics and plant breeding that we have incredible tools to make new crops. We can we can breed 3 times faster, or 15 times faster than we could in the last century. And so really I think it's the question, what do we do with this powerful technology for society and the globe? And I know around here, of course, and why I really enjoy being here at Cornell is that there's lots of people thinking about these questions. And of course, we're working in these areas, trying to do more with less in terms of fertilizer and water, doing some work on biofortification. And then my current pet project that I really want to get off the ground is perennializing maize. And then I think we should replicate that in 100 crops. So that would be the subject of a different talk. But what I want to end with is that I've really had a great group of colleagues the last decade who've collaborated and really put this all together. And so if you're interested in NAM, these are the PIs that helped put together, and Nick Lepak, who's here, is locally is the guy who really made it happen on the field and everything. And then I think I mentioned a lot of the other folks as we've kind of gone through the talk. So I'd be happy to take some questions.
Well, I have a question. So this intergenic variation, is that mostly in promoter region, so that would be farther away from the gene? And so then I have a follow-up question. Comment on the challenge of once you move to from analyzing the genome to biochemical variation, comment on the challenge of associating this variation with a specific gene, like how often it's going to be the gene that's directly next door or the...
Right. No, I think the challenge of figuring out, let's say, you get there's an enhancer element in between 2 genes. Which gene does it affect? Or does it even, is it many away? I think some of the nicest data that's now coming out on that. Well, first off, when people have done positional cloning, quite often in maize, the controlling elements are 60 kb, 80 kb away, okay? And those are the ones people were able to make sense of, okay? I don't know how many times they probably people started out the experiment and just never could make sense of it. But I think if you look at the ENCODE results that have just come out in humans, there is connections between chromatin. I saw one plot that at 120 kb apart, there was essentially an enhancement of those regions of the genome coming together. I see no reason why that isn't totally common in our genetics. And we have proven examples of enhancer elements that are now close to a megabase away. So I have no doubt chromatin structure that emanates over many millions of base pairs could be affecting the expression. And so you may clone one thing, it may make no sense to you, but it makes genomic sense if you knew what portions of the genome touch one another. Okay, anything else? Okay, great.
Oh, there's one question.
Yeah, I'm talking about the rare allele. And can you give some comments about like a category, for example, present absent, that type of rare allele, absentee rare allele, for example, two statters or three statters, how these?
Yeah, so I don't, the distribution of rare, so I would first say I don't think we have a really good feel for what the allele frequency distribution of rare alleles are in intergenic regions. It will likely be different because the mutational processes, actually Votek may be a little bit better at addressing the relative mutation rate of single nucleotide changes versus how often are we getting an unequal crossing over or a slippage there or some kind of excision event that may cause some of these intergenic regions. I mean, there are estimates of these rates. Right now, I don't until the intergenic ones, I don't really believe it until we can sequence larger fragments. So I always have my fingers crossed that the nanopore, something that'll give us 5 or 50 kb of sequence will come out, and it will, whether or not it happens next year or 2 years from now or something like that. But I think once we get to that scale, then we'll have an accurate estimate of what's going on and the rate of those intergenic regions. So.
Given the model of pseudo-predominance, which informs everything I know as well, do you predict that within 10 to 20 years we'll have inbred maize that will perform as well as your hybrids? Will we undo the business model of the private sector? Because it has huge implications for rice, where we don't have the inbreeding depression, where hybrids are being driven against all biological reality, but there is still this interesting question, right, about the hybrid versus inbred.
So and still a little bit from Cinta and Sarah's work that they'll be talking about. Sarah's not here, but essentially, we've made a heck of a lot of hybrids recently, and there's nothing in there that suggests there's any big steps in heterosis. Everything is lots of small, tons of little load, or modest load. And so I think right now, in the long term, I don't think we can do it with normal crossing over. I think at some level it needs to be done with allele replacement.
Is that a strategy for increasing recombination instead of rare regions?
No, but I think there's some Arabidopsis folks who've come up with some clever genes to really change things. So I think those are certainly worth trying. But I mean,
I was going to say, Wojtek was coming up with that.
Have you come up with that?
My answer's good.
I think it's a topic for another seminar.
I think increasing paracentromeric recombination is certainly a worthwhile goal. But we also can just genotype till we can find them. I mean, it's not that hard to genotype. If you were a multi-billion-dollar seed company, you could catch those crossovers. But I still think, let's say genetic load is made up of 5,000 load loci per haploid genome. You can't recombine out those fast enough. So it needs to be allele replacement, which I think is doable. And I think the question is, when does it technically become doable? Okay. Thanks.